Hi, this is Miss Litton, and this is AP Bio Unit 2, Essay Multiple Choice Review. I want to remind you, to look at your Master Hyperdoc. If you are from a different school, that Master Hyperdoc can be found on my home page for the school. And you want to look at these for the big ticket review items. This is like if you wanted a study guide, these lists are your study guides on what's important. When I go through and write tests, I literally read through these and go and find questions that support these things. Okay, so that's how you want to approach it. All right, so this whole thing was cell communication and the cell cycle. Wait, is that right? Oh no, I put 4 on there. That is a Master Hyperdoc, but which Master Hyperdoc do we want? 2. Did that scare you for a minute? Scared me too. So we want, chapter or Unit 2. I just gave you the wrong one. Unit 2 Master Hyperdoc. Okay, and what does that have on the top of it? Some cells probably. Okay, so the first thing on this one was the structure of a cell, the size of a cell, that's what we started off with, and the cell theory. So do you remember the parts? Oh my God, Karen. Do we remember the parts of the cell theory? All living things are composed of cells. Then what? Cells are the structural and functional unit of all organisms, and cells, what? Only come from pre-existing cells. What is that process if you're prokaryotic? Binary fission. If you're doing it for growth and repair, what might it be? Mitosis. If you're doing it for sexual reproduction, it might be meiosis. Good. All right. This is on the wrong side. Okay. We then talked a bit, and I know this page is screwed up, but we're trying to understand why cells were small, and the reason why cells are small is they need a large surface area per volume. Because what happens across the cell membrane? All exchange of materials, right? And remember, how does one cell talk to another cell? It can either secrete a chemical or direct contact. We saw that in the immune system, right, when we studied the immune system. Okay. All right, and then we talked about problems related to surface area and homeostasis, issues with, when we look at the elephant, why do they have these big flappy ears? What are they using them for? Cool down. Yeah, to cool down, to exchange their blood supply, the temperature in their blood versus the environment to cool down. But yet this whale is trying to keep things very, very what? Compact, close to his body because he's what? Trying to conserve heat because it's a mammal. Okay, so we can look at structure and function related to homeostasis. Homeostasis, that's a new word. And there was something else I can't remember. I give up. Okay, we looked at the 3 domains. And the first domain that we talked about was bacteria, and we went over the different structures in bacteria. Let's start from the center and work our way out. Do they have a nucleus? No, what do they have instead? A nucleoid region where their DNA is kept. Can you tell me something about their DNA? Circular. A single circular chromosome. Are they diploid? No, they're not. They do not have 2 copies of their chromosome like you do, right? We're starting to learn about that right now, about dominant versus what? Recessive. Because they just have 1 single circular chromosome. Is that the only DNA they have? No. What else could they have? Plasmids. They could have plasmids, exactly. Extra rings of DNA. What's something they have and all living things have, and we talked about this as a conserved property? Ribosomes. What happens on ribosomes? Protein synthesis. And that's where you use what to build what? Messenger RNA to build polypeptides. Another name for that are proteins. Okay, we wouldn't say amino acids because that's just an individual unit, right? We are building proteins that are made up of amino acids. All right. Something else. What do we call the thing that goes around the outside? The first thing that goes around the outside. Cell membrane, right? So all cells have cell membrane. Now, whether you may or may not have a cell what outside of that? Wall. Cell wall. You may or may not have, you'll have different types of carbohydrates. Like we have a glycocalyx, but no cell wall. So we have some external carbohydrates, but they could have one that is very hard, that could be protective, or it could be kind of slimy in that way. But that is your glycocalyx as well. What are all these tiny little hairs things? What are those called? Not cilia. Fimbriae. What is that for? Attachment. What is this? There's a pilus. What's that pilus for? Right. It's a sex pilus for exchange of DNA. All right. Are these cells smaller or larger than eukaryotic cells? Smaller. They are smaller. I want to continue. Okay, good. All right. Off here to the right, I have archaea. Very, very similar, that they are what? What's the similar part? They're prokaryotic, except they tend to live in what? Extreme environments. But archaea, are they older or newer than bacteria? Older. Newer. They have that, they were misnamed, right? They were misnamed as ancient bacteria, but it turns out they're very closely related to our kinds of cells. What are our cells? Eukaryotes. Yeah, good. All right, here I just wanted to show you a plasmid. Here we can see the 3 domains. How did cells go from being prokaryotic to eukaryotic potentially? Yep, that's what it's called, endosymbiotic hypothesis. But what happened? Are you seeing one prokaryotic cell moved into another prokaryotic cell? Okay, all right. Can you give me some evidence to support that hypothesis? Of what? The energy organelles. What are the 2 energy organelles? Mitochondria and chloroplasts. And the idea there is that mitochondria and chloroplasts were free-living bacteria that went to go live in another bacterium. And our evidence about that is both mitochondria and chloroplasts have their own DNA. They do have ribosomes that are the same. Have their own DNA, and the DNA is what? Circular. As opposed in eukaryotic cells, what kind of DNA do you have? Linear. So that kind of makes sense. They have their own ribosomes. They can make their own proteins on those ribosomes, and they are self-replicating. And what was the other one? I heard somebody say it early on. Double membrane, right? So it looks like they have all the characteristics of a living organism that would live independently as a prokaryote. And they're just living inside of a eukaryote. Now, you don't have to worry about your mitochondria abandoning your cells, right? Because they have given up all those control mechanisms to the central nucleus, right? So they don't have the ability to leave at this point. Mitochondria and chloroplasts have that double membrane, and that kind of helps us understand how we could get a nucleoid region to become a what? Nucleus. And how could that have come about? Invagination. Great work, okay? So the cell membrane folds in on itself, and then that indent of the cell membrane wraps around the nucleus to become the nuclear envelope. And then what's supportive on that is that the nuclear envelope doesn't have one membrane, it has 2. That would make sense if it folded in on itself, yes? And then the entire endomembrane system, and we know the endoplasmic reticulum is continuous with the nucleus, right? Which would make sense if it was just an extension of that ribbon of membrane that folded in, yes? So we have the endoplasmic reticulum. What's connected to the rough and smooth ER? What's directly connected to that? Golgi apparatus. They all kind of work together, right? Okay, so that would be another piece of evidence. Then your next part that you need to do for this section is just know your organelles. Know the structures, know the functions, be able to recognize them, be able to recognize how they relate to one another. Like, how does the nucleus relate to the ER, which relates to the Golgi, right? Maybe you would add in ribosomes in there, maybe somewhere in that little discussion. Okay, so how do they relate to one another? How do they help to maintain homeostasis? What are similarities between an animal cell and a plant cell? What are differences? You could very easily Google, a labeled plant cell or a labeled animal cell. You probably already have it in your group shared notes from when we talked about it, and you could review all of those processes. I know you know that the nucleus is where the DNA is, right? And the DNA does not stand alone. It is wrapped on those histone what? Proteins. Good. And you only see it in the shape of a chromosome when it's getting ready to do mitosis or meiosis. Okay, then it'll condense into those rod-shaped structures, but otherwise you just see it inside the nucleus. But there's still some dark kind of sphere inside the nucleus. What is that called? Nucleolus. And what is that busy making? Ribosomes, yeah. The rRNA that's part of ribosomes, exactly. All right, coming off of the nucleus, we have the rough ER, and the ER is kind of like a little highway through the cell. So what's going on in that compartment is different than what's going on in the cytosol outside of the cell. And if it's rough ER, it's rough because it has what on it? Ribosomes. It has ribosomes on it. And the rough ER transitions into what? Smooth ER. And they have different functions. On the rough ER, those ribosomes are there, so of course that's where you're going to do some protein synthesis. But there are free-floating ribosomes that can also have protein.
Please excuse the interruption.
No,
Andrew Brandama, please report to the office right away. Thank you.
Okay, but proteins, when they are made on the ribosomes on the rough ER, will move into the interior of the endoplasmic reticulum. They will pass through the ER, could go from the rough ER into the smooth ER, or directly out and packaged in a little membrane that's coming off of the rough or smooth ER, and then it'll transition to who? Where does it go next? Golgi apparatus, which we think of as the UPS man, because he processes, packages, and does the final mailing on there to determine is it going to stay inside the cell or is the cell going to secrete it. Okay, and that's your endomembrane system. Now there's some other packaged proteins inside the cell. One, the stomach of the cell, what is that called? Lysosome. So it contains hydrolytic enzyme. It has enzymes to break things down, right? Let's see. I think that's good on that. What do you use to store things potentially? Vacuoles. Okay. What's something an animal cell has that a plant cell doesn't have? Centrioles. That's one thing. You know what the mitochondria do, powerhouse of the cell. You know what chloroplasts do, right? Okay. So cytoskeleton. So the cytoskeleton has different size filaments, and remember, a cytoskeleton is one of those things that is a process that's conserved in what? Yeah, all cells. And so you have to have a cell move around, and the cytoskeleton is going to help you do that. We talked about centrioles already, 9 groups of 3 microtubules. We could look at some kind of evolutionary connections because cilia and flagella do not have 9 groups of 3. They have 9 groups of 2 with 2 singles in the middle. But the thing that anchors the cilia and flagella, what's that called? Basal body. And the basal bodies were back to 9 groups of 3. So there's some connection there, right? Between centrioles and basal bodies for sure. And then a modification for cilia and flagella. All right? And then, like I said, last but not least, you want to make sure you find a diagram maybe similar to that, okay, to review the different structures and functions. I want to look right here. So on the essays, cell organelle structures and functions most critical and why. So remember I asked some of you that. I don't remember who I asked that. What would you say are the most critical ones, and why would you say that? If you left off the nucleus, that tells me you don't have a good understanding of the cell, right? What would you think is another really critical organelle? Mitochondria. Absolutely, okay? So pick things that are critical to the functioning of that cell. Could you tell the difference between prokaryotic and eukaryotic cells and write to me about that? Yes? And how eukaryotic cells evolve from prokaryotic cells. Could you talk about the endosymbiotic hypothesis? All right, create a model or a diagram to show the path of protein in a eukaryotic cell from its formation to the secretion. We went over that as well. All right, And so next, let's go on to the next one, next chapter. I'm so annoyed that I put the wrong hyperdocument on. Okay, so the next thing was cell membrane. So take a look at some of the essays and let's see if we address this. The structure of the cell membrane, protein functions, 3 ways to cross the membrane, and active versus passive transport. You could just sing your way through that entire chapter. You remember everything about that, right? So let's start off with structure. 4 major components in the structure of a cell membrane. 2 are the biggest ones. There's one main one. It's called a bilayer. A what? Phospholipid bilayer. So you better lead with that, phospholipid. And there's interesting properties of being a phospholipid because we know a phospholipid is a modification of a what? A lipid. Okay, what kind of lipid? A fat, right? Lipid has 2 subcategories. What are they? Fats and steroids. So a phospholipid is a modification of a fat. A fat normally has glycerol and 3 fatty acid chains, yes? Okay, but a phospholipids, you've taken one of those fatty acid chains away and you've replaced it with a phosphate group. A phosphate group has phosphorus and oxygen. We know about oxygen, right? So the phosphate group would be hydrophilic, and that's because it could interact with what? Water very readily, right? Whereas the fatty acid chains, we know these are carbon-hydrogen chains, a hydrocarbon tail, so that's going to be hydrophobic. So we look at the arrangement, it makes sense. Hydrophobic here in the center, those of the phospholipid bilayer, and then hydrophilic here on the edge. I see something floating in there. What is that? Cholesterol, exactly. Cholesterol helps with the what of the cell? Fluidity, right? It helps if it's too cold that it won't freeze, and also so it won't just melt away or fall apart if it's too hot. Okay, so it is a fluid mosaic layer. What's the mosaic of it? The proteins. So the proteins, some of those proteins span the whole entire membrane. Those would be called what? Integral or integral, yes, because they're going from one side to the other. If they're only on an edge, they are called what? Peripheral. Then we see that when we look at both sides of the cell membrane, it is not the same on either side. Because to the interior, what do we have? Cytoskeleton. To the exterior, what do we have? Glycocalyx, right? We have glycolipids and glycoproteins. They are all, glycolipids and glycoproteins are made out of the same stuff. What are they made out of? Sugars. Okay? And that chain of sugars is attached to a lipid if it is a phospholipid, and it's attached to a what? Protein if it's a glycoprotein. I made up a new word. Okay. Glycolipid, glycoprotein. Sorry, I was still laughing at something I had overheard. Okay, good. So then next, fluid mosaic. We hit that? Good. So let's talk about protein functions. I know you know them. Here we go. They could be a channel, carrier, cell recognition, receptor, enzymatic junction. At this point in your young little life, okay, you should have examples for all of those, right? Could be a channel, could be a carrier, passive active, receptors, right? MHC1s, MHC2s, BCRs, right? There's so many things there that we could pick, right? Cell recognition, enzymatic, what are we thinking about there? Enzymes are embedded maybe to trigger something like glucose production. Okay, so those are all things that we could be thinking about in that region. How do we get across a cell membrane? Well, we could go through the phospholipid bilayer, but not if we are large or charged. We could use a channel or a carrier or endocytosis or exocytosis, which would be the whole membrane, yeah? So on this through the phospholipid bilayer, what regularly goes through the phospholipid bilayer? Water, oxygen, carbon dioxide. Okay, water, oxygen, and carbon dioxide. Okay, most readily. If you had a cell wall, is the cell wall going to keep those three things out? No, plants would be dead if they did, right? Bacteria would be dead if they did, right? So cell walls are not going to prevent that. Cell walls are not so much about determining who goes in and who goes out. They're more about what? Structure. They are to help plants when plants are in a environment. What do you think? Talk about it right now with somebody next to you. Water must flow from the hypo. If they are in a hypotonic environment, water will flow in and that plant will expand and expand. Not just expand, they would burst, like our red blood cells would, right? That's why I can't jam her with a syringe of distilled water. Well, for many reasons, but that is one reason. Okay? Because that would make all of her red blood cells do what? Burst. That's why if I'm going to jam her with a syringe, I want it to be a what? What? Isotonic solution. That's when you go, focus here. That's when you go and get an IV at the hospital, or something like that. That's an isotonic solution. Same to your red blood cells. What if I jammed, what if I jammed her with like salt water, seawater? What kind of solution would she be in? Hypertonic. Water must flow from the hypo. Water would leave her body cells and go out into the solution, yes? Okay, that's how you kill a snail with salt, right? Right? That's, remember when the whole pickle thing? That's the whole pickle thing. All right. Next. Hang in there, guys. Hang in there. You're doing a good thing right now because you're reviewing, right? When you're reviewing, stop for a minute. Your presence here is just one step, right? What makes this effective for you is when in our discussions, things that we identify, red flags go off, okay? And you're like, oh my gosh, I got to review that, right? And you're jotting that down. You know, it's not the sky is not painted red right now. There's a lot of stuff I know because you've been telling it to me right now. But where you want to focus your efforts on things that I say where you're like, oh gosh, I totally forgot that. Then go back and look at it, right? Okay, so let's talk about this. Passive versus active transport. Passive transport, can that happen through a protein? Yes, what is it called? Facilitated diffusion. The protein is facilitating the process of something going from a what? Higher concentration to a lower concentration. Why is it having to use a protein? It's either what? Large or charged. Okay? But passive transport, as long as it's O2, CO2, water, they can go right from a higher concentration to a lower concentration. What's something that facilitates water transmission across a cell membrane? Aquaporins. Good. Okay? Active transport is something that requires what? Energy. You're going against the concentration. You're going from a lower concentration and moving something that's at a low concentration and moving it into an area where there is a higher concentration. That takes energy to move something like that. But that's a great tool. You can use that difference in concentration to do work. Maybe like make some... Exactly, because that energy was used, right? That's your proton pump. That's when you are taking protons and moving them from one side of a membrane to the other side, because then they're highly motivated to go back through passively. And you're going to capture that energy when they move back through to make some ATP. Okay? Active transport is not just using a protein or through the electron transport chain, but it could be whole membrane, which would be what? Endo and exocytosis. Good. All right. Signal transduction. We remember that, and I bring it up here because this is an area where we learn about what? Receptors. We learned about receptors. That's one of the protein functions. And your receptor could be on the surface of the cell or it could be internally. Right? transduce, that signal could have other signals that need to be there. Could these signals ever be misinterpreted? Yes, but they will end up having some sort of cellular response. Okay, osmosis we just talked about, so I bet you feel good about that, right? When you look at this, side B over here, how would you, oh, it's labeled, darn it. What is side B? It is what? Hypertonic. Why? Because it has more osmotically active substances in it. What do we call those osmotically active substances? Solute. And the water is the solvent. Good. So water is going to flow from a higher or lower water potential? Higher water potential to a lower water potential. Okay? All right. And we talked, I'm going to skip that right there. So we have, we talked about isotonic solutions, hypotonic, hypertonic. We already discussed that. Our famous pickle. And then this brings us to water potential, which we also had a lab on, right? Which we just started talking about. We talked about an open jar of water, no pressure on it other than atmospheric pressure. Okay, the best water potential, maximum water potential you could ever have is what? 0. And then as soon as you start adding stuff into it, then it becomes what? Negative. The water potential goes negative. So when you take a cell and put it in there, let's assume that that cell has stuff in it, right? If it has stuff in it, it's not 0, right? And in this case, they calculated it, negative ICRT, yeah? You could calculate the solute potential, right? And we know it to be -2. You know how to calculate that because you've done a lab on how to calculate that, right? So if they calculate it to be -2 and you put it in this distilled water, the water is 0, right? That's a higher water potential. This cell is -2, which is a lower. Water is going to flow from a higher water potential to a lower water potential. And it's going to fill up that plant cell, but only to the point where the turgor pressure, right, starts to push back. Because remember, it's a factor, is it right here? It's in another slide I have. Your water potential is a factor of your solute potential plus your pressure potential, right? So if my solute potential right here is -2, water is going to go in until the cell wall is pushing back just as much to +2, and then -2 and +2 equals what? 0, which is the water potential of the distilled water. You okay with that? That guards all movement. Do we have cell walls? So do we have turgor pressure? No, we don't have a pushing back mechanism. Yes? Louder.
Can water potential ever be positive?
No. I mean, unless you manufactured it due to a pressure situation, you know, but no. Just best water potential, you can get a 0. Now, your pressure potential could be positive. There could be issues with, or sorry, negative. There could be like a reaction like that where there's a pulling on it to draw the water into it, so that is a possibility. Okay, moving on. We talked about using a protein. We talked active transport, like what's the most famous pump we have? The sodium potassium pump. And we talked about endocytosis and exocytosis. Good. And then what was that one? Receptor-mediated endocytosis. Good. This one. Yeah. Are we okay? All right. Then we just very briefly said there are different junctions. You could have proteins that are continuous between one cell and another. You could have channels that are continuous. You could have cytoskeletons that are continuous in order to help animal cells stick together because animal cells do not have cell walls. Good. All right. Then that brings us to your lab that you did in this unit. Okay. Remember this was... We had the first part of the lab where we just saw diffusion. I skipped over. It's here. It's here. Okay? In this part of the lab, remember we put starch and glucose inside the dialysis tubing, and it appeared what color? Clear, right? Then out surrounding it, we had a, let me get a pen here somehow, crayon. Okay, out here in the water, what did we put? We put iodine out here in the water, which was kind of that orangey brown color, right? And then we set it in the bag. We set the bag in the iodine, and it was clear. But then what happened to the bag? It turned what? Blue. What did that tell us when the bag turned blue? Iodine had what? Moved into the bag, and who did it react with? Starch. When iodine and starch mix, we checked it before we did the experiment. When you mix iodine and starch together, it turns blue. Now, what moved? You told me the iodine moved into the bag. How come you didn't tell me that the starch moved out of the bag? Yeah, if the outside turned blue, that would mean that the starch could get out. But it didn't, so it couldn't. How did we test whether glucose could move? Glucose test strip. I mean, they could give you a solution like on the exam. There's something called Benedict's solution. I don't expect you to memorize that. You have to boil it. It turns different colors if there's so there's other testing solutions besides glucose test strips. But we can, all you need to know is that you can analyze for the presence of glucose, right? Okay. Then the next part of the lab, we did different molarities of sucrose inside of our bag. And remember, we hypothesized that if I had unknown solutions, I could ask you, hey, what do you think this solution is? What do you think this solution? And the way you would base that is the percent change in what? Mass. Because you would put your whatever, your 0.2 molar sucrose solution in your bag. You would leave room for that in that bag to expand, because you know if it's a 0.2 molar in here and this is distilled water, water will flow from a higher water potential to a lower. And by having the sugar in here, it's got a lower water potential. Yes? Okay, so water must flow from the hypo. The water would flow into the bag, and we would expect after some period of time, 30 minutes, that the bag would weigh more. Okay, but if instead of a 0.2 molar solution in there, I had put a 0.8 molar solution in there, relative to the 0.2 molar solution, do you expect that bag after 30 minutes to weigh more or less than the 0.2 molar? More. Because it is even more hypertonic, right? So water is really motivated. Water must flow from the hypo. So even more water would flow into that one, and that bag would have even more weight. And all you would have to do is use a graph and plot out the percent change in mass, final minus initial over initial. You would plot that out on there, draw a line, and you would be able to identify, right, which molarity is what. Would you agree with me on that? Just by whoever changed the most mass. Okay, then we did the next thing. We took, remember our cylinders of potato? Okay, and we did not, no, look at me, take a breath in, let it out. Focus on understanding this lab right now. Some of you are falling asleep. I know you've had a long day and you're getting ready for finals and you have a ton of pressure, but just stay here on this last thing, okay? That potato, it had an unknown molarity. So what did we do? We took chunks of potato. Remember the potato core? Remember that? We took those potatoes and we weighed them. We said, this is the initial weight. Then you took chunks of potato and you put them in various solutions. I think each team had 3 solutions they were in charge of, yeah? And so then you went to see, hey, how did the potato weight change? In some of the solutions, they gained weight, and in some of the solutions, they lost weight, okay? What were we trying to find? We were trying to predict, infer from our data, what solution we could put it in where it would neither gain nor lose. Because at that point, there would be no what? No pressure. And if there was no pressure, then to calculate the water potential, we could use that negative ICRT to calculate the solute potential. Okay, if I went too fast there, let me see, I have something, I think. Yes. So we had this graph. We had the different molarities, right? 0 would be distilled water, 0.2, 0.4, 0.6, 0.8. You would predict any potato that is put in 0 molarity, right, distilled water, would it gain or lose mass? It would gain, right? Because that water has greater water potential than the potato who has stuff in it. So it's going to gain some sort of weight, right? And so we thought if we could plot that, maybe at 0.2 it doesn't gain as much weight. At 0.4 it starts losing weight, and then more weight, and then more weight somewhere in there. And then we would draw a best made line through here, yes? Do you remember that part? And then we would look to go, well, we didn't nail the solution exactly, but we were able to plot it. And when I go straight down here, I am then inferring from my data that at 0.3, let's say, molar, it would neither gain nor lose weight. Okay? And once you do that, then you can use this equation right here. Because water potential equals solute potential plus pressure potential. If we find an area where it neither gains nor loses weight, we can get rid of pressure potential. So then we have a formula for solute potential. It is equal to negative ICRT. Okay? What does the T stand for? Temperature in what? Kelvin, right? Temperature in Kelvin. And so you'll have this on your equation sheet when you take the AP exam. So the temperature in Kelvin, you just take degrees Celsius plus 273. What is R? Yeah, it's a constant. It's right here. So you just plug in that 0.0831 liters per bar per moles per Kelvin, yes? Okay, then what's this C? Yes, the C is where we were right here. It's this molarity where it did not gain or lose weight. Okay, and then what's this I? Ionization. Glucose, sugar, does not ionize, so your ionization constant is 1. If you did it something salt does ionize into what? Sodium and chlorine, or chloride, yeah. And then it's negative. It's just a negative in front of that. So from that, okay, you're just calculating then what the water potential is. Just simply tell me this. If I have a carrot that I've kept in a cup of water versus a carrot that sat on this desk for 24 hours, who's going to have more water potential? The one in the water, right? This carrot that sat on a dried out old mangly carrot, right, has less water in it, right? So it would have a lower water potential. So just make sure you have your big picture painted, and that should help you. Yes?
Will we have the equation sheet on our final?
Absolutely.
Okay.
Okay, last thing I want to address is we introduced the nervous system versus the endocrine system, the speed, and we use that as an example of transmission of information. So exocrine glands are like sweat glands. Endocrine glands are what secrete your hormones for you, like your pituitary gland and your thyroid gland, right? Your adrenal gland, testes, ovaries, those are all glands. And we talked about protein hormones versus steroid hormones. Do you remember that? Because steroid hormones like testosterone and estrogen and those hormones from your adrenal cortex, those are all lipid-based, those steroid hormones, so they can go right in and impact the DNA. They have long-lasting effects, whereas protein hormones have to stand at the door and knock. Remember, they have a receptor and then they can signal something inside. And we talked about comparing the nervous system with the endocrine system. The nervous system is faster than the endocrine system. And we talked about feedback, positive feedback, I think, right? Or was this a different unit? Yeah. Positive feedback is something like giving birth, right? It's amplifying in one direction until some culminating event, whereas negative feedback is you're trying to maintain, let's say, a temperature or homeostasis at one set point. I think that's it. Hopefully you feel better now than when you walked in. And when you go out, you want to reflect, what is it that you need to work a little bit more on to feel comfortable for your final? Okay? All right, good job, guys.